Good day, this is Professor Will, CBMD PhD. Today, Saturday, September 26, 2020. It is 2:37:41 seconds a.m. Eastern Standard Time, and this is a continuation of my microbiological research, particularly gene transfer. So let's hop to it here. It might seem that horizontal gene transfer, HGT, would be a complicating factor in understanding how the various major cell types evolved. Yet, far from being a complicating factor, HGT turns out to be the essence of the process. As is now well known, HGT appears to have had, at certain times in evolution, the potential not only to introduce entirely new functions into cells, but to displace any constituent gene in the cell with an alien equivalent. What effect has HGT had on the genetic record of organismal descent? What is its relationship to canonical pattern? Understanding here pivots on what the characteristics of HGT are and what factors determine them. The interested reader will find a more extensive and useful discussion of the major problem of evolving cellular organization and its relevance to the universal phylogenetic tree structure. The most obvious thing about HGT is its extreme variability in both frequencies and phylogenetic range, from one gene to another, from one major taxon to another, and likely from one evolutionary stage to another. It turns out that the dominant factor shaping HGT is the organization design of the potential recipient cell. The various components of a cell are in one way or another, to one extent or another, interconnected, variously integrated into the cell's design fabric. Some of them are weakly and simply integrated into the fabric, others tightly and complexly so. Components of the first type are in effect modular. Functionality and structurally they are pretty much self-contained and self-defining, i.e. their interactions with the rest of the cell componentry are minimal. It stands to reason that such a modular, loosely coupled component is far more likely to undergo horizontal gene displacement by some alien equivalent than is highly and idiosyncratically integrated componentry. An ideal system for accessing the character of HGT is a translation apparatus, for while most of its componentry, e.g. the ribosomal proteins and elongation factors, are tightly coupled into the rest of the mechanism, and hence the cell fabric, the tRNA charging enzymes, aminoacyl, tRNA, synthetase are not. The latter are modular, are loosely coupled into the cellular fabric. And as might then be expected, the ribosomal proteins and elongation factors have been largely unaffected by HGT, whereas the aminoacyl tRNA synthase have been quite highly buffeted by it, both in terms of the frequency which HGT has affected them and the broad phylogenetic range of many of the alien displacements that have occurred. It is common, for example, to see the archaeal genera of a given aminoacyl tRNA synthase in one or a few eubacterial lineages. A detailed phylogenetic analysis of 20-odd aminoacyl tRNA synthase shows 1. That about 2/3 of the tRNA charging enzymes exhibit the canonical pattern typical of the other components of the cellular information processing systems, and 2. That in all these cases, the canonical pattern has to one extent to another been eroded by HGT. But 3. That the degree in which and the ways in which that pattern has been eroded are idiosyncratic, qualitatively different for each of the enzymes. From these facts it was concluded that the canonical phylogenetic pattern itself was not produced by HGT, but reflects some earlier evolutionary dynamic. Thus the canonical pattern represents partial genetic record of organismal histories that extends back to the root of the universal tree. In other words, while HGT has been a powerful, pervasive, and shaping force in organismal evolution, especially early on, it has not completely obliterated the ancestral organismal trace that the universal tree represents. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and computer science. I hope you enjoyed listening. Until next time, have a great day.